Right, hello and welcome back to the channel. I haven't made a video in a while, but I've had some requests, so I thought I'd make one. So in this video, I'm going to discuss the required practical 4, which is investigation into the effect of a named variable on the permeability of cell surface membrane, since this one was requested by a few people. And this is for AQA A Level Biology. So first, I thought I'd give you just a bit of background into the knowledge behind the practical. So before watching this video, I would recommend that you are somewhat knowledgeable about the transport across cell membranes topic, which I will link in the corner because I have a video on that, so you can watch that if you haven't covered that in class yet or you're not confident on that knowledge. I'll give you a little bit of background. So a cell surface membrane, so that's the membrane that surrounds a cell. It's made up of something called a phospholipid bilayer. And that term phospholipid bilayer is really, really important to know because it comes up all the time in exams. So yeah, the phospholipid bilayer makes up cell surface membranes and they are selectively permeable. So yeah, I've highlighted that term in red because that is also really important to remember. So selectively permeable, that basically means that it only allows certain things in and out. So it's selectively permeable. And this permeability changes with variables. So there are so many variables, but some of them, so the main ones, they're pH, temperature, solvent concentration, things like that. So yeah, this practical we're investigating permeability. And for this practical, we use beetroot cells, and the reason why is because, as you know, beetroots are like purple or red, and this means that it's really easy to spot changes. So that's one of the main reasons why we use beetroot for this practical. So how you kind of spot the changes in this practical, so if the beetroot cells are more permeable, then more pigment leaks out. And this pigment is called betalain, so that's the red pigment or purple pigment that you see in beetroot. And you measure the permeability by the amount of pigment that leaks out. And you measure this amount by measuring the light absorbance. So if there's more pigment, then obviously less light would be able to go through it. So that means that permeability is higher. So in terms of the method, so first of all, using a scalpel, you would get your beetroot, you would obviously have to cut it into cubes, probably about 6 to 10 cubes. I mean, it depends on how many repeats you're wanting to do. But the really important thing to remember is that these cubes need to be identical in shape, because obviously that would affect the amount of pigment that could come out. And it's also really important before you proceed with the experiment that you blot or wipe the excess pigment that's come about from slicing the beetroot into the cubes, because you don't want to mistake that pigment for pigment that has come out as a result of the experiment. So the named variables that you can investigate, the main ones are solvent concentration and temperature. So I'm going to briefly go through all of those. So if you're investigating solvent concentration, you use ethanol for this. So the first thing that you want to do is to create different ethanol concentrations using something called a serial dilution. So this term serial dilution again is another really important term. It's not just a dilution, it's a serial dilution. That means you take one tube and then you take some of the liquid out of that tube into another tube, and then some of the liquid from that tube into another tube, and so on and so on. So the solution will gradually get weaker and weaker. So if you're asked about this in an exam, they wouldn't give you a mark if you just said dilution. You'd have to say serial dilution. So for the purpose of this video, I'm going to do this as you're investigating 100% ethanol to 0% ethanol. Right, so for this, you can use as many concentrations as you want, but for the purpose of this video, we're just going to use 5. So let's just imagine that these tubes are, so the one on the left is tube 1, and then tube 2, tube 3, tube 4, and tube 5. So let's say we want 10 milliliters of liquid in each of the test tubes. So the 100% ethanol tube, so that's tube 1, obviously 100% ethanol, so that's just pure ethanol, so you just want 10 milliliters of ethanol in that tube. And then the 0% ethanol, obviously there's no ethanol in there, so we just use water in this case. So in the 0% ethanol tube, so in this video's case it will be tube 5, you'd want 10 milliliters of distilled water. And then for the rest, so 2, 3 and 4, you want 5 milliliters of distilled water in those. So to dilute them then, so to dilute to the 100% ethanol to say 50% ethanol, what you would do is that you would take 5 milliliters from that 10 milliliters of ethanol and add it into that 5 milliliters of water. And then you get the 50% ethanol. And then you just repeat that process. So once you've mixed that 5 milliliters of ethanol and 5 milliliters of water, you take 5 milliliters of that and put it into the next tube. So as you can imagine, it's getting weaker and weaker as you're doing it. And the resulting concentrations are, so obviously your first one's 100% ethanol, and then the next one's 50%, because you've taken half of that volume. And then that's for each tube it's reducing by half, so the next one's 25% and the next one's 12.5%. So if you're investigating temperature, it's a bit more simple. So you place your cubes of beetroot into volumes of water in test tubes. Obviously it's important that the volumes of water are the same. And you place your test tubes into water baths of different temperatures. It's up to you what temperatures you use, but usually it's like 10, 20, 30, 40, 50, 60, or whatever you want. So the next steps are obviously suitable for both variables, so your ethanol concentration and your temperature. So you want to wait for about 20 minutes. And then what you want to do is you want to filter your samples into a cuvette. So by filtering the samples, I mean you just want the liquid, you don't want the cube of beetroot, because you're investigating the pigment that's come out. So you just want the liquid, and you want to pour it into a cuvette, which is that you'll probably have used them before, those glass cuboids. You then want to measure the absorbance of all of your cuvettes. Obviously each cuvette has a different tube in it, obviously. So to measure the absorbance, you want to use something called a colorimeter. So when you're using your colorimeter, you want to set it to the blue filter. And then you want to calibrate it to 0 using a cuvette with pure distilled water. You can probably use if you're doing the ethanol concentration one, you can just use that 0% ethanol cuvette. But it's really important that you calibrate the colorimeter so that it's as accurate as it can be. And then obviously for each solution, so if you're doing your temperature, it will be the 10, 20, 30, 40, 50 degrees one. And if you're doing the ethanol concentration, it would be the 0%, 50%, 25% one. So for each of these, you need to measure the absorbance and then plot the graph against either the concentration or the temperature. So yeah, the risk assessment. So you might be asked in an exam what the risks are, but this is mainly for your practical endorsement. So obviously the first hazard is the ethanol, because ethanol, it's an alcohol, so it's very, very flammable. And it's also an irritant as well if it gets on your skin. So the control measure against this is that you obviously have to keep it away from naked flames. I mean, you don't use flames in this practical anyway, but it's just a precaution. And also it's important to wear goggles in case it gets in your eyes. And then the next hazard, if you're measuring temperature, is the high temperatures of the water baths. So that provides a risk of burns or scalding. So the control measure for this is to use tongs to remove your tubes from the water baths, and also to just generally handle it with care and keep the tubes away from the edge of your lab bench, because the tubes could roll off and splash and hurt you. And obviously with most practicals the glass is a hazard, and also the scalpel used to slice the beetroot. So the risk there would be cuts. So also the control measure for this is to just generally handle it with care and also keep away from the edge of the bench, because if you have a tube that falls off it could break and then damage you. So the general findings and conclusions from this practical then. So a higher absorbance indicates a more permeable membrane, because obviously there will be a higher concentration of pigment in the solution. So if there's a higher concentration of pigment, less light can pass through, so that means that it will be absorbing more light instead of just letting it through. So yeah, higher absorbance indicates more permeable membrane, as there will be a higher concentration of pigment in the solution. So in terms of temperature and what effect that has and why, so absorbance increases with temperature, and the reason for this is that heat. Well, if you have done the proteins and enzymes topic, then you will know that heat, if you go above a certain temperature, it denatures proteins because it breaks bonds in the tertiary structure. And this is the same for membrane proteins, as you will know that membranes have proteins in them. So yeah, absorbance increases with temperature as heat denatures bonds in the tertiary structure of membrane proteins. And this denaturing of the membrane proteins obviously means that the membrane proteins can't function. So this means that the membrane is more permeable, because the membrane proteins control what comes in and out. So if they're denatured, it will make the membrane more permeable. And also a reason why the membrane's less permeable at low temperatures is because the phospholipid bilayer is very rigid at low temperatures. And this is because it's kind of like if you've looked at kind of particle models in previous years, you'll know that solids, obviously they're solids because the particles are packed together. Obviously when they're heated up, obviously they can melt and become a liquid, because the particles kind of are less packed and they can move about. And this is kind of the same principle with the phospholipid bilayer. So the phospholipid bilayer is rigid at low temperatures as the molecules pack together, and this causes a decreased membrane permeability. With the ethanol concentration, it kind of has a similar pattern to temperature. So as the concentration increases, the ethanol will disrupt the cell surface membrane, because it's an alcohol. Alcohols denature proteins too. So the ethanol will disrupt the cell surface membrane structure more. So this will lead to membrane permeability increase, so more betalain is released, so absorbance increases. So that's kind of it for the content. So I thought I'd go through a few exam questions that are related to this topic. So we'll go through these now. So I'll just read them out. So this question, so the cells of beetroot contain a red pigment. A student investigated the effect of temperature on the loss of red pigment from beetroot. he put discs cut from beetroot into tubes containing water. He maintained each tube at a different temperature. At 25 minutes, he measured the percentage of light passing through the water in each tube, so that's your absorbance. So the question then, so the student put the same volume of water in each tube. To explain why it was important that he controlled this experimental variable. So at this point, you get these questions a lot. So why this variable is there, why this one's here, why this one's not, why it's really important that this variable's there. So here's what I've put. So if the volume of water was different, then obviously that would affect like the concentration of pigment, obviously, because there's more or less water, that would affect the pigment. So for example, if they put in too much water, so more than the others, then the solution obviously will appear lighter because it will have diluted a bit more because there's more water. And obviously this would mean that the absorbance would be less, even though it so for that particular temperature or whatever, the absorbance was supposed to be more, it might be less in this case if they put in too much water. So yeah, having the same volume of water in each tube ensures that the results can be compared. Okay, so part B then, so describe a method the student could have used to monitor the temperature of the water in each tube. So this is a 1-mark question, so it's very simple. Sometimes some students can get caught out with these kinds of questions because they seem too simple. So monitoring temperature, so just think about what you use to monitor temperature. So it's basically a thermometer, isn't it? So all you need to put really is that a thermometer is used to take temperature readings. So yeah, use a thermometer to take temperature readings. Let's look at the mark scheme. So for part A, the first marking point, so it says if there's too much water, the concentration of pigment will be lower, or it will appear lighter, or the more light passes through than expected. So this is what I've said, so we'd get that mark. Or you can put the opposite, so if they put in too little water, then the concentration will appear darker and it will be greater, so the less light will pass through, so the absorbance might be higher than it should be. So marking point 2, so that's your explanation. So this is kind of a general point if you're asked about a control variable. So obviously the reasons for control variables are so that the results can be comparable. And it says here, ignore reference to too much water, so red pigment or solution is too weak to measure. So that's not really acceptable because it's not really scientific enough, and it's not using the vital terms like light passing through, absorbance and things like that. So you wouldn't get a mark if you put that. So part B then, so it says, so 1-mark question, so there's only 1 marking point and it's a simple question really, so there's nothing else that you can write. So take readings during the experiment using a digital thermometer or a temperature sensor. So the terms take during the experiment and digital are in brackets, so you do not need to use those terms to get the mark, but you can do. And it says readings, so the letter S there is in bold and it's underlined, and that's because it's vital that you've understood that multiple readings are being taken, because it says monitoring the temperature in each tube. So that says that you're taking multiple readings. So you need to make that plural in order to get the mark. So the next part then, so part C, so you've got a graph here that shows the student's results. So you've got the temperature of water in degrees Celsius on the x-axis and percentage of light passing through the water on the y-axis. So it says draw a suitable curve on the graph above. So there's 2 ways that you can do this really. So you can do a line of best fit, or you can just do it like a line graph. I've just done a curve like this. So part D then, so the last part. So the decrease in the percentage of light passing through the water between 25 degrees Celsius and 60 degrees Celsius is caused by the release of the red pigment from cells of the beetroot. So it suggests how the increase in temperature of the water caused the release of the red pigment. So we've just mentioned this. So I've written here, increasing the temperature damages the cell membrane, increasing fluidity in the phospholipid bilayer. This is kind of your set answer if you're writing something like this. So I haven't said denaturing the membrane proteins. I think you can write this, but it's not. There's nowhere in the question that mentions membrane proteins, so you can write an answer that's a bit more general like mine. But if the question, like the text in any part of the question is mentioning membrane proteins, then you would need to mention membrane proteins in your answer. That's another exam tip for you there. And obviously your explanation, so that increases the fluidity in the phospholipid bilayer. So the reason why I've not put it increases the permeability is because it doesn't say suggest and explain. So if it said suggest and explain, I'd say that increasing the temperature damages the cell membrane, so the membrane is more permeable. It just says suggest and it's 2 marks, so I've kind of put 2 different things there. So increasing the temperature damages the cell membrane, increasing fluidity in the phospholipid bilayer. So the mark scheme then, so part C, so the curve. So you can do like a smooth curve, kind of like a line of best fit, or like something like drawn like a line graph, so point to point, drawn between the points. So it doesn't really matter which one you use because, well, you're not really measuring something over time, so it doesn't matter. So you can do either, but I would do a curve. It's just kind of easier. So it says reject any extrapolations below 20 degrees or above 80 degrees. And this is really important because you haven't measured any of these temperatures, so you don't really know what the results would be. And yeah, so you shouldn't extrapolate. So that means kind of carrying the line this way or that way. It says reject. So when a question says reject, that means if you put that, you don't get any marks at all for the question, no matter what else you've put. And also your line should look smooth, so not messy or sketchy, like the mark scheme says. So yeah, part D. So there's 3 marking points in this one, but it says 2 max. So if you put all 3 of them, you're not going to get 3 marks. You just get a maximum of 2. So the first point is about damage to the membrane. Or you can write about the membrane proteins denaturing, but it's not absolutely essential. And then there's a point about increasing fluidity in the phospholipid bilayer or damage to the phospholipid bilayer. Right, so that is all I want to say for this video. If you've got any requests for videos, please leave them in the comments, or if you have any questions, please ask. And I will see you in my next